Okay, let's get started. You only have to put up with me for an hour and 5 minutes more. Yay. Or something like that, unless you're taking class from me next term. When is the final? Tuesday. What time? 8 o'clock. Yes, so be here for that. I won't be. I have to give a seminar at the University of Florida. Poor me. But my graduate student will be here and proctoring that. I will be close to my cell phone if there are any really, really problematic issues that happen at that time. So any questions about the exam at this point? Multiple choice. I am going to be concentrating more on the stuff that I've covered in my lectures rather than those that we had in the guest lectures. So there'll be a little bit more of that, but certainly that material will be on the exam. And that's why I'm going to be reviewing it specifically today. Are there other questions about the exam? 50 questions, multiple choice, not cumulative. Okay, we're all happy. We're all going to get 50 points. Yes?
When you say that you're focusing more on what you go over in lecture, like the exams we had the guest lectures, do you mean specifically what we go over in like today's class or this session, or that you're still pulling from material from their lectures but just more?
Okay, so the question for those of you out in internet land, it really has to do with am I going to be just taking questions from what I covered today or from those whole lectures? From those whole lectures is the answer. So not necessarily what I have pulled out today, because it's 4 lectures that I'm trying to do in 50 minutes. Other questions? Okay, and as promised, we also have some clicker questions today. So our first clicker question for today is, why does one perform a yeast-2 hybrid screen to detect protein DNA interactions, to determine which DNA sequence a transcriptional activator binds to see if a protein has activator activity, to detect protein-protein interactions, to see if a DNA sequence can function as an enhancer? Thirty seconds. Ten. So most people seem to think it has to do with detecting protein-protein interactions. I agree with you, so that's a good thing. We are going to talk a little bit today about some of the other assays that are used to look at protein-DNA interactions and specificity of those DNA interactions as well. So good. Everyone's going to do well on the exam, hopefully. Our next question. Imprinting is due to histone methylation, histone acetylation, DNA methylation, scaffold protein binding, or phosphorylation. 30 seconds. Ten. Let's look at the results. Yay, everybody seems to agree that it has something to do with methylation, and most people seem to think it has to do with DNA methylation, which is correct. Histone methylation is often on the way to getting DNA methylation. We'll talk a little bit more about that today. Histone acetylation is mostly important for what? So for gene activation. And so again, we'll talk more about that a little bit more today. Scaffold protein binding. In some cases, the imprinting causes differences in scaffold protein binding. And so that was the example that I talked about last time, is that one specific example. And phosphorylation, I needed another answer for, make 4 of them. So let's actually go through this here again. Sorry about that. Let's get this right so people actually get their points. I did realize why some people were getting 4 points, even though we only had 3 questions. And that is that you actually get points for voting and showing up at all, because I misconfigured things, but I'm going to leave it that way because it's too much of a pain to change it. So, and I figured that people would appreciate it. Oh, do you want me to go through and take away all those points? No. Okay, good. I won't. So in our last clicker question of the term, before they bind to the messenger RNA, CPSF and CTSF are free in the cytoplasm, free in the nucleus, bound to poly-A polymerase, bound to the poly-A binding protein, bound to the CTD of the RNA polymerase. Thirty seconds. Ten. So there's slightly more of a difference of opinion on this, but most people seem to agree it's the binding to the CTD of the RNA polymerase. The first question here, of course, is what the heck is CPSF and CSTF? These are doing what? Important for poly-A tail formation. So they cleave the messenger RNA and give the start site for the poly-A polymerase to extend those cleaved messenger RNAs. And so that's the first part of the question. So if you've got that, then the rest of course, has to do with where are they beforehand. And if you think about it, that's where you're going to get poly-A tails. It's also the beginning of termination of transcription in eukaryotic messenger RNAs. And so it's really advantageous to have those proteins right where they need to be when you're going to get termination. And so they are brought along for the ride along with that C-terminal domain of the RNA polymerase. And I don't know if Dr. Bartlett talked about this, but my favorite analogy is the tool belt of RNA polymerase. That's the CTD. It's got all kinds. It's the splicing factors are associated with it, the capping factors are associated with it, etc. So everything that it needs is right there when that RNA is actually being made. So those of you who need to take off can quietly get up and leave. And now we will continue talking about the rest of the material that was covered on those guest lectures. Really sort of, 4 lectures. Main one, first one anyway, talking mostly about DNA binding and the DNA binding proteins, the motifs, how you get DNA binding, and the methods for detecting DNA binding. Then our second guest lecture was about epigenetics and how you get inheritance in the absence of some kind of change in your sequence. That, of course, has to do with DNA methylation, chromatin remodeling. And when chromatin remodeling, I take this as being a very general case where you also have all the modifications that happen to the histones. And then the Polycomb proteins, which are really critical in terms of these non-genetic changes to gene expression. And particularly important is this connection between the Polycomb proteins and a lot of these small RNAs, or the non-coding RNAs. A little bit about messenger RNA processing. We talked about tailing last time. That was sort of filling in at the end of that lecture. So we'll talk a little bit more about capping and splicing. And then the small RNAs, that was Monday's lecture, either microRNAs, small interfering RNAs. And if we get a chance, and there's still a couple minutes at the end, if I talk fast enough and you don't ask enough questions, I'll talk a little bit about ribosomal RNA processing. That won't actually be on the exam, but I think it's important stuff to know. Regulation of gene expression. So this is basically sort of the main theme, and it should be, of this whole course, and to some extent, molecular biology in general. If you look at what our genetic material is, it's DNA. What does most of the activity, of course there's now RNA that does some of this, are these active proteins all the way over here. And you notice that there's multiple different possible levels that this amount of active protein can be regulated. Either at the transcriptional level, where you're making the messenger RNA, processing that messenger RNA is another level you can control it, getting that RNA out of the nucleus, getting it to the right place is another way to control it, how much of it you have around, how much you translate it. You can control the activity, and this is particularly important with your binding of various different ligands, phosphorylation, or you can also degrade the protein. So these are really the different levels. We talked about all of them, but probably the most important of them are right here at the beginning or right here at the end. And the reason for that is that transcriptional control, if you control transcription, you don't have to do all the rest of that stuff downstream in terms of control. And so it's a very high-level control. At the very near end, where the active or inactive protein is concerned, this can be a very rapid kind of regulation of activity, but you've still spent a lot of energy in making your RNA transcripts, splicing them, etc. So this is not very energy efficient. This is much more energy efficient. This is relatively slow compared to changing the activity over here. And most of gene regulation, as far as we can tell, really is at this transcriptional control level, particularly if you're thinking about development and cell biology aspects of things. So those transcriptional regulators are interacting, of course, with the DNA, because that's that first step. These are all of the gene regulatory proteins. They bind to double-stranded DNA, and that double-stranded DNA, it's really important that you've got the major groove in particular that has sequence-specific information. And so you bind to that major groove in the absence of having to unwind the DNA. These gene regulatory proteins bind in a sequence-specific manner, so you get them associated with some regulatory region in your DNA, and they're binary. They're either bound or they're not bound. And when they're bound, they'll have an inactivity or not have an activity. And so if you think about fine-tuning exactly how much gene expression you're going to have, that's going to be due to the combination of different factors that are there, not one single factor having a range of particular activity. So we know a lot about how these proteins interact with DNA, mostly from bacterial systems and particularly viral systems. How many of you are taking virology next term? See you again. This is bacteriophage 434, which we actually won't talk about, really similar to the lambda repressor, where you have this classic example of an alpha helix binding in the major groove of DNA, and alpha helices, all their side chains stick out around the outside, so those are then going to be able to interact directly with the base pairs. Another nice example here is this is a dimeric protein. This is the identical amino acid sequence up here as it is down here, but they're in different orientations. Hopefully you can see that this beta strand here is coming out in the front in this monomer, and it's coming out in the back in that monomer. So they're flipped over relative to each other, and that means that they're binding to what kind of sequence? Inverted repeat sequence, yes, because this alpha helix is exactly the same as this alpha helix. So those amino acids sticking out on the outside are going to be exactly the same. But if they're flipped over relative to each other, they're going to have to bind to inverted repeat sequences. That's the particular helix sitting in the major groove. Again, it's a very common theme. There are exceptions, but pretty much all of the DNA binding proteins that we talk about in this class are going to have binding of an alpha helix in the major groove. There are a couple of different ways of doing that. One, let's actually back up here really quickly, is what's called the helix-turn helix. And so that helix here in the major groove has another helix on top of it. So helix turn, which is in the back here in the yellow one, helix. There are also so-called zinc finger proteins. These, again, are also having an alpha helix sticking in the major groove of DNA. These are some of the very few DNA binding proteins that you can predict just from looking at the primary sequence that it's going to have a DNA binding motif because it's got 4 amino acid residues, either 4 cysteines or 2 cysteines and 2 histidines, which are actually going to bind to zinc and give this particular structure. And again, it's an alpha helix which sits down in the major groove. There are 2 different kinds of these zinc finger proteins. There's this one, which is 2 cysteines and 2 histidines. Usually these form 2 beta strands and an alpha helix. This is a quick aside. It's a reminder. This is a motif. This generally is not stable by itself. It has to be in the context of a larger domain. So it's one of these sequences that gives you a particular function. Again, this is DNA binding. The other kind of zinc finger is actually more like a helix-turn-helix motif. Here's that zinc bound here. But again, it's putting a alpha helix in the major groove of DNA. So helix-turn-helix, and then the sort of eukaryotic equivalent of the helix-turn-helix, also known as the homeodomain, which has, since it says a domain, actually has that full structure, but it's basically as a helix-turn-helix sticking that helix in the major groove of DNA. Another kind of DNA binding motif, which is pretty common in eukaryotic DNA binding proteins, is the basic helix attached to some kind of dimerization domain. Example here is AP1, where you have a leucine zipper dimerization domain, and then 2 DNA binding domains down here at the bottom. NFAT, nuclear factor AT, don't worry about the name here, but this is one of those examples where it actually doesn't have an alpha helix sitting down in the major groove of DNA. But what's important about this slide and why I pulled it out has to do with why you want to dimerize, why you want to have multiple different proteins coming together in order to bind to DNA. Each of these individual DNA binding proteins, AP1 or NFAT, can't bind to DNA very well by itself. So NFAT can't bind this, AP1 can't bind this, unless the 2 of them are together. And if the 2 of them are together, then you have really strong DNA binding. And it's because when they interact with each other, particularly this piece right here, that helps both of them to bind to the DNA. This gives you what's called cooperative binding, so individually not very much, together very strong. And this is partly why people do all those yeast 2 hybrid screen assays, for instance, looking for protein-protein interactions. So those protein-protein interactions are at least as important as a lot of the protein-DNA interactions. Talk a little bit about dimerization motifs already. This, again, is that coiled coil or leucine zipper dimerization motif next to the basic helix. You also have these helix-loop helix dimerization motifs, a helix-loop helix, not to be confused with helix-turn helix. Again, I don't like the terminology, but that's what we have. The helix-loop helix is a dimerization motif, whereas a helix-turn helix is a DNA binding motif. Just to keep those two separate from each other. Another great thing about dimers, as well as getting that cooperative binding, is it also allows you, or you being the cell, to have a lot more flexibility in terms of what DNA sites these particular DNA binding proteins interact with. And if you think about human genome, 3 billion base pairs in each of your haploid genomes, so 6 billion base pairs, you've got to find out exactly the right place to bind at the right time and place. So having these multiple different proteins, each of which binds to a specific sequence, and so this is just the example up here of a basic leucine zipper, or basic coiled coil protein. Each of these different color greens, dark green binds to blue, light green binds to red, and so you can mix and match all of these. Just 2 proteins can bind to 3 different sequences. And the more of these things you have that can dimerize with each other, the more different sequences you can actually have. These can be very similar proteins to each other. They can also be very different proteins relative to each other. These are 2 homeodomain proteins with this helix-turn-helix motif, helix-turn-helix motif. Or you can even have 2 domains that are attached in the same protein, binding as if they were dimers. But they're 2 different sequences of proteins binding to 2 different DNA sequences. So this is Homo and Heterodimers. And again, the homodimer means you've got 2 of the same. Heterodimer means you've just got 2 different ones. And again, just allows you many more possibilities of how and what DNA sequences you can bind to. As if that wasn't complicated enough, this is what a normal promoter or gene regulatory sequence would look like, bound to multiple different proteins, some of these heterodimers, and it's this combination of proteins and the combination of binding sites that you have down here, which allows you really fine-tuned regulation. Some cells are going to be activating some genes at some times, etc. So each individual protein, again, is on or off, but it's the combination of all these proteins together that will lead to expression or not expression of a particular gene at a particular time. So this is what's going on inside the cell. It's very difficult to piece apart exactly which of these things are going on at any given time. So now we'll talk a little bit about some of the techniques on how to do that. The first one is either called EMSA. They don't like very much. Most people will call it the gel shift assay. But EMSA stands for electrophoretic, let me get rid of this, gel mobility shift assay. And basically all that it is you have a DNA fragment, and these are just multiple copies of that same DNA fragment that you mix together with, say, all those proteins that you saw on that last slide. Then when you run them out, you separate those fragments on a polyacrylamide gel, and those fragments that have bound to a particular protein are going to go through that gel either faster or slower. Nothing bound is going to run the fastest. Each of the individual proteins bound is going to run a little bit more differently. And so here, in this cartoon case, we've got 6 different DNA binding proteins, all of them of different size, all binding to this one piece of DNA. You want to separate them out relative to each other. Now you do column chromatography over here, and you can actually separate each of these proteins and test them with this gel shift, electrophoretic mobility shift assay. So here in our first fraction, which is fraction 11, here at increasing salt concentrations relative to your column, this is an ion exchange column, here you have proteins C2 and C3 that bind to this particular piece of DNA. So you can separate those, you can get the mass spec of that protein, you can figure out what it is, and you say this binds to this fragment. Now, complex, or your binding protein 1, etc. And this process of separating out these individual DNA binding proteins is exactly how people came up with things like TF2A, B, C, and D. You know, one of these would be A, one would be C, one would be D, etc. And so it's just literally by the separation and looking for DNA binding at particular promoter sequences. This tells you that a protein binds to a piece of DNA, but it doesn't tell you exactly where. It tells you it binds on that piece of DNA, but not exactly where it is. And to figure that out, you have to do an assay like DNA footprinting. And the whole idea with DNA footprinting is that you have a whole population of different DNA molecules, all of them labeled at one end, that are different lengths from each other because you've put in something that chops up that DNA. And so in this case right here, in the absence of a DNA binding protein, you're going to get a piece of DNA which is cut here, piece cut here, piece cut here, piece cut here, piece cut here, and piece cut there. And so you can have a whole population of different DNAs with slightly different lengths relative to each other. If, however, you have a DNA binding protein that binds to this piece of DNA, it will stop this cut here, but you'll have pieces of DNA that are cut on the outside of where that protein is interacting with DNA, and that gives you the so-called footprint. What you're looking for is absence of cutting in one particular region. And so that's shown right here. In this case, we have no protein bound, and then the TATA binding protein bound. And you see here, there's no cleavage that's taken place, no piece of DNA of this length when you have the TATA binding protein present. And this turns out, TF2D binds in a whole bunch of different places, including downstream of the promoter. Here's a nice example right here. If you put TF2D into your complex at plus 20 relative to no protein, you see quite a lot of binding downstream of that. So that's a DNA footprinting assay. It's relatively difficult to do. I spent years of my life trying to do these things and get them to look pretty. Usually it's about 10 before you get one of these that looks this nice. What people are doing more and more these days to get an idea of what DNA a particular protein is binding to is chromatin immunoprecipitation. And this is also really nice because you can now see inside a cell how you have binding of a particular protein to a particular DNA sequence. DNA's footprinting, you have to start with one particular DNA sequence and see where your protein's bound to it. Whereas here, it could be all of them inside the cell. So people are going much more in this direction. So the first step here is you cross-link the DNA to your proteins. There's chemical ways of doing this. Won't get into the details here. Once you have this cross-linking that's taking place, then you chop up this DNA into lots of small pieces. So now you've got lots of small pieces of DNA, some of which are cross-linked to proteins. Now you just care about one of those proteins, so you put in an antibody to YFP, your favorite protein. And that way you can separate the protein, now bound to the DNA, from everything else which is in there. All the pieces of DNA that are not bound to protein, all the pieces of DNA that are bound to other proteins, doesn't matter, they're all gone. Now what you can do is reverse this cross-link and then sequence this piece of DNA. And that tells you, again, what specific DNA sequence your favorite protein is bound to inside the cell. So there are questions about DNA binding and assays and things. Otherwise, we'll move along to epigenetic processes. So there are really 4 different ways you can get, again, epigenetic inheritance is inheritance either in a cell or sometimes even in an organism from non-genetic changes. So a non-sequence change in the DNA which is still inherited. And one of the ways to have that is just a positive feedback loop. So if you produce a protein, for instance, a transcriptional activator, in a cell, which regulates itself, then as soon as you have cell division, that transcriptional activator will still be there. It will activate the expression of its own gene, which will make more of the transcriptional activator, et cetera. So any kind of positive feedback loop can give you epigenetic inheritance. Another way you can get epigenetic inheritance, and this is we've talked about quite a bit, is histone modification, things like telomeres, centromeres, anywhere where you have heterochromatin. This actually serves as a positive feedback loop as well, because we talked about a lot of those histone modifying enzymes, which will then recruit histone modifying enzymes, which will modify the histones, which will recruit another histone modifying enzyme, et cetera. So whatever your histone modification is, that gets maintained, again, mostly through these positive feedback loops. DNA methylation is exactly the same way. DNA methyltransferases bind to methylated DNA and will methylate DNA right next to where you already have methylated DNA. This is very important for gene repression. We talked about this for X inactivation last time, also for imprinting. And so you can actually keep that particular region. Now, of course, if you have these positive feedback loops, there's one obvious worry that you have. How do they not get completely out of control? How do you end up with a positive feedback loop that stops? And so stopping these positive feedback loops is really important as well. And then the last case that I don't know if this was covered in much detail by Eric Iverson, but you can also have a change in the state of your protein if it's a autocatalytic event. And so this is what happens with prions and prion diseases, and potentially in some of the neurodegenerative diseases like Alzheimer's or Parkinson's, is that you have again, it's another positive feedback loop, but now it's a protein aggregation process which goes on. DNA methylation, hopefully we're not beating the dead horse on here. 5-methylcytosine, this is what happens in eukaryotic DNA. Other methylations happen in bacterial and archaeal DNA. But here's this 5-methylcytosine. Doesn't change your base pairing, but is going to provide a mark to say, hey, this is a particularly important region of your DNA, and this only happens in CG sequences. So not CG base pairs, but CG sequences from 5' to 3'. The reason that this is so important is once you've started that methylation, again, it leads to a positive feedback loop. That's just described here. Here we have starting out with a modification of your histones. These modified histones will then bind to DNA methylases. Once you have this DNA methylase which binds to your modified histone, it will methylate all of your DNA through here, and then these DNA methyl binding proteins will compact the DNA. And so this is classic what's happening in heterochromatin. It's also to some extent what's happening, we'll talk a little bit more about the details there, with X chromosome inactivation as well. So that's these histone modifying enzymes, also known as histone writer complexes. Another way that you can change your chromatin structure is through the so-called chromatin remodeling enzymes. These are ATPases, which literally will move DNA along histones. And so the example here, which is unfortunately a little hard to see, but this DNA here basically gets pulled into this complex, and this piece of DNA comes back out. And so you can see how this is really important for instance, allowing access to a TATA box or some promoter sequence, or even a enhancer where you're going to get some of these transcriptional activators that are being bound to it. So these chromatin remodeling complexes can move your DNA relative to nucleosomes. In some cases, they could even pop the nucleosomes off completely. But it's an ATP-dependent process that you have to use for this. Hopefully, again, this is beating the dead horse a little bit. Histone modifications clearly lead to lots of changes in chromatin structure. So if you've got acetylated histones, particularly lysine 9 in histone H3. That usually leads to opening up of the chromatin. Very common that you see these associated with transcriptionally active genes. So this is euchromatin. On the other hand, if you get methylation, and particularly methylation again at that 9 or sometimes the 7 position in histone H3, that leads to heterochromatin formation. So it's really the modification on the histones, which seems to be sort of the first step in making the decision. We're going to be euchromatin or heterochromatin. The acetylation process is a relatively rapid one, acetylation and deacetylation. Histone methylation seems to take a little bit longer. So rapid changes, so genes on, genes off, usually acetylation. If you're going to be turning genes off for longer periods of time, it's usually a methylation process. If you look at some of these modifications, and I mentioned already histone H3, lysine 9, when this is acetylated, very often that acetylation process happens through the activity of an enhancer binding protein. And so here's this gene activator protein right here. That will bind to a histone acetyltransferase, and in many cases these histone acetyltransferases are also known as co-activators. I don't know, hopefully Dr. Bartlett talked a little bit about this, or maybe Iverson as well. The whole idea of a co-activator is it's a non-DNA binding protein that still stimulates transcription. And basically the way that that happens is through, again, protein-protein interactions between a DNA binding protein and then this active or activation part. And this particular activation here, acetylation at lysine 9 and histone H3, leads to binding by TF2D. So TF2D binds here, and TF2D also will recruit other chromatin remodeling complexes to get more and more access to that one particular piece of DNA. So this is sort of a transcriptional activation process. First thing that happens, you have your gene activator protein, then your co-activator will methylate. Once you have methylation, you also have kinase activity. It's that kinase activity which leads to binding by TF2D, which then also helps getting your chromatin remodeling complex and eventually give you transcription. So going from this case, potentially even a case of heterochromatin, to transcriptional activation. What does these? This is mostly the so-called SIR proteins, SIR for the silent information regulatory proteins. Those SIR proteins were all found in yeast mating type regulation, because in yeast cells you've got the two copies, one of A, one of alpha, two different sexes of your yeast cell, which are silenced. And they're only, once they get transferred to the mat locus, or the mating type locus, then they're active. And so it's in yeast mutants where they found that we were getting too much activation, where a lot of these proteins were found. Turns out that these SIR proteins are almost always histone deacetylases. So histone deacetylase, what's that going to do? It's going to take acetyl groups off of your histones and generally lead to silencing, because hypoacetylated, so low acetylation, leads to much less of your chromatin opening up. And that's basically just shown here in this process. There are multiple proteins here involved, lots of different SIR proteins, and it's not critical what each of those names are. Another way of looking at this is here, activation up here at the top. Repressor at the top, activation down here at the bottom. Activation where you have histone acetyltransferases through this co-activator protein. You put on acetyl groups, you open up the DNA, Tata boxes are accessible, you can get transcription. The flip side of that is when you have a silencer sequence, which binds to a repressor with a repression domain. This binds to a co-repressor. So you've got co-activators and co-repressors. This co-repressor is a histone deacetylase, which will take those acetyl groups off. And again, taking the acetyl groups off, the title box is not going to be accessible. We're not going to have access to it. So this is what you have, really rapid regulation. Again, histone acetylation and deacetylation. You also can have the longer-term process, which is the methylation leading to much more heterochromatin formation. Most of that seems to happen through these Polycomb complex proteins, where you have these basically starting as a co-repressor, but then they'll recruit histone methyltransferases. You methylate each of these histones. That will bind to other Polycomb complexes, which will also stimulate DNA methylation and completely compact your DNA. And you can actually see that down here, where you have Polycomb complex together with nucleosomes. And hopefully, again, they mentioned this, but Polycomb were mutants that were found in Drosophila, that when you had a mutation in Polycomb, you ended up with expression of too many genes in other places. And that leads, of course, to, okay, if you have a mutation, expression of too many genes, that means that protein is probably involved in repressing genes when they should be repressed. So that's how these were originally found. So there are questions about epigenetic inheritance before we move on to any of the RNA processing. RNA processing, basically, everything you need to know and more is on this slide. First thing that happens as soon as you have transcribed a particular piece of DNA, you make this RNA. It immediately gets capped. The capping enzymes are associated with our tool belt, the C-terminal domain of RNA polymerase II. Then, while again you're transcribing, you get splicing that takes place, and then polyadenylation. This then, if it's happened properly, gets exported from the nucleus and translated. There are many places where you can get regulation. Here, splicing hasn't happened properly, polyadenylation hasn't happened properly. You chew up the RNA before it even has a chance to get outside of the cell. And then you also have regulation here of these already made messenger RNAs through microRNAs and some other processes. Also regulation of the amount of RNA that you have in the cytoplasm with lots of exonucleases. So exonucleases, again, these are nucleases that chew in at the ends of nucleic acid structures, either being DNA or RNA. In this case, these are all RNA exonucleases. Again, same process here. Here's your DNA. Reds are exons, blues are introns. Transcribe, this is what the RNA polymerase makes. As it's making it, you make a cap. Then you have these introns, which get removed. And in fact, introns get removed usually before you have polyadenylation. And then this ends up getting exported. Let's talk about the cap structure. There are 3 enzymes here. The RNA polymerase makes a transcript that starts with a triphosphate. So again, this is very different than DNA polymerases, which can't start by themselves. This triphosphate, the first of those 3 phosphates gets removed by a phosphatase enzyme. And then my favorite enzyme, guanylyl transferase, try saying that one 15 times quickly. Guanylyl transferase takes a GTP and flips it over and attaches it on the other end. And so this is a very strange 5' 5' bond that you get between the guanine, which gets put onto the end of your cap structure, and then the 2 phosphates that are left from the original part of your transcript. And it's probably because of that this is now protected from all those exonuclease activities, which will be coming in at the end, because they can't recognize that particular structure, and provides a very specific place for your cap-binding enzymes, or I say cap-binding proteins, which are important for export and protection to actually associate with. Most cases, you also then have a methyltransferase, which is going to add methyl groups to both the guanine and the riboses, which are present in the actual transcript. And we'll see some more examples of that in virology again next term. And that's just shown over here in a little bit more detail, just a slightly different way of looking at it. So that's your cap structure. Splicing, originally shown in virus viruses. And this is probably one of the most famous splicing micrographs. Most people don't realize it's from a viral RNA. Here, this is the adenovirus hexon gene, which has, as usual for most of these genes, really big introns and relatively small exons. So again, the red here are exons and the blues are introns. And the way that this was shown is people isolated the messenger RNA here, which you can see in red. Well, it's the interpretation of this micrograph. Must have stared at this for a really long time to figure out this. You can see that this particular piece here is slightly thicker than that one right there. So this is your DNA plus RNA, and that's just your DNA by itself. So you hybridize these things together, you see these big loops. And so this big loop represents an intron, this loop represents an intron, and that loop represents an intron. What's this extra stuff at the end here, probably? Just RNA? What do you put at the 3' end of RNAs? Poly A tails. Is that going to bind to the DNA? No, it's not going to bind to the DNA. So this is that extra poly A tail just hanging out at the end here. So this shows that splicing takes place. First, people thought it was just in viruses, but it turns out it happens in cellular RNAs as well. How do you get splicing to take place? It's due to these sequences which are present at the ends of exons and the beginnings and middle of introns. One of the big problems with understanding splicing is that these consensus sequences, again, you line up a whole bunch of DNA sequences and then you try and figure out what things are well conserved. Well, it turns out that very little is conserved in these sequences. And you've got really long introns. And so it's actually kind of hard to predict where you're going to get splicing that actually takes place. So GU and AG, but you find these all throughout all kinds of different sequences. So you have to have other ways of figuring this out. There's also a highly conserved adenine residue right here, which is where you get the strange 5' 2' interaction with the spliceosome complex. How do you detect where these things are? It's all through RNA. So RNA is a really critical component of the splicing machinery because it, through RNA-RNA base pairing interactions, tells the spliceosomal machinery where your intron and exon boundaries are. So here, the U1 small nuclear RNA will base pair at the 3' end of one exon. U2 actually binds both at the branch point and here at exon 2 and brings these things together. If you look at how this seems to be taking place, U1 binds again what's also called the splice donor site, so the 3' end of the first exon. U2 binds to this A in the middle, but also the splicing acceptor site, which is here at the 5' end of the next exon. These undergo all kinds of funky rearrangements, and those funky rearrangements all have to do with base pairing, those base pairs being pulled apart and then being put back together. So you actually need a lot of helicase activity, which is taking double strands apart and putting them back together in terms of getting your spliceosome to work. You have this end, which is created by probably the activity of the U6 SN RNA, which then will cleave. That then gets hooked up to the 2' OH, which is on the adenine residue in the middle of the intron, and then this free OH at the 3' end of the first exon gets attached to the this 5' end of the second intron, giving this lariat structure, which again gets cut out and is removed. So, just quickly again, U1 base pairs, U2 binds the branch point, U4, U5, U6 make the whole spliceosome complex, rearrangements, and again, all these rearrangements are RNA base pairing interactions. Then U6 and U2 give you esterification, transesterification. Again, that's making this lariat structure. And then you join up the two exons together. How do you know, again, to do these things? The consensus sequence is not very well conserved. So to do this, you need many other proteins, and those are particularly the SR proteins. And just SR because they've got lots of serines and arginines in each of these proteins. So these proteins bind specifically to exon sequences, and they're also usually called exon splice enhancers. So the particular sequence in the exon will bind to one of these SR proteins, and those SR proteins then form bridges together with these snRNPs. So the presence of an SR protein then stimulates the interaction of these snRNPs to get splicing to take place. So, of course, the next question is, well, what's regulating these SR proteins? And so that leads to the alternative splicing processes, and the alternative splicing that can take place is due to the activity mostly of these SR proteins. The spliceosome is basically the same for everything. It's just which of the exons those guys interact with, either at the donor or the acceptor site, 3' end or 5' end. Those are what determines which of the pieces of RNA are included. And the massive, sort of the extreme in terms of alternative splicing is this Drosophila DS-CAM gene, where you have potentially 38,000, actually more than 38,000 different possibilities in terms of the splicing that can take place from one gene. So you've got 14,000 genes in the genome, but up to 38,000. So 14,000 by itself doesn't sound like very many. 14,000 times 38,000 is a heck of a lot. And now, of course, this is the extreme case. There are a couple of other things I wanted to mention here really quickly, and that is that there are only a couple of places where you have alternative splicing in this gene. All the gray exons are always used. And so the vast majority of the exons, this is true for almost all alternative splicing, these guys are all included. It's just a few things which are alternatively spliced. And so, again, in this case, we've got 4 regions that are alternatively spliced, 12 here, 48 here, 33 here, and 2 here. And the way you get 38,016 is you multiply 12 by 48 by 33 by 2. So pretty straightforward. One nice example of how alternative splicing takes place and is regulated is through studying of sex determination in fruit flies. This actually was happening downstairs in the lab where I was doing my PhD. So we had our seminar speakers have a lunch and our snacks afterwards, and you see a fruit fly is floating around. One of the students go, "Hey, that's one of mine. That's a sex comb reduced," or "That one is a sex lethal mutant." So this is how a lot of the splicing determination was found. And so what happens in the fruit fly, your X to autosome ratio is 0.5. In the female, it's 1, all dependent on the presence of this sex lethal protein. Sex lethal is a splicing regulator. So what sex lethal does is it blocks the splice acceptor site for one of the introns in both its own gene, so it serves as a positive feedback loop, but more importantly, in this transformer gene. So the transformer gene, as true with all of these mutants, if you have a mutation in that gene, it's non-functional, you end up transforming males to females and females to males. But in the normal case, in the transformer gene, this is now a splicing stimulator, which, absence of it, you get splicing that doesn't have this exon. You have the final double-sex protein with one exon, which is male-specific. If you have the activation of the transformer gene, you get splicing that gives you a female-specific gene. So it's first a splicing repressor, that's sex lethal, which then regulates a splicing activator, double sex, which will then lead, transformer, and then which leads to the activity of double sex, which is really what leads to this transformation. It's basically another way of showing it. I wanted to give you a second slide to take a look at that. So there are questions about mRNA processing. Otherwise, we'll talk really briefly about small RNAs. A couple of different things that happen with small RNAs. The first one is attenuation through a riboswitch. And the riboswitch is basically a RNA sequence which is present at the 5' end, in most cases, some cases the 3' end, of your messenger RNA that binds to a specific ligand. So it's not protein binding, it's the RNA itself binding to a particular ligand. And that binding can either cause a secondary structure to form which gives you transcriptional termination, or it can also regulate translation, usually again by forming a secondary structure which blocks your ribosome binding site. This is the case in bacteria because you've got coupled transcription and translation, so that can happen that way. There are a few riboswitches that you find in eukaryotes, but they're much more common in bacteria. On the other hand, the small RNAs, the microRNAs, and the siRNAs, these are the ones that are much more commonly found in eukaryotes. So there are two different kinds of these. The microRNAs, these are really endogenous. When I say endogenous, this is something which you normally have in the cell. It's normally being transcribed. MicroRNAs are being made by RNA polymerase II, just like messenger RNAs. These are short RNAs that give you a secondary structure once they've been transcribed. The secondary structure is giving you double-stranded RNA. That double-stranded RNA gets exported into the cytoplasm, it's processed, and then has some kind of activity. Let's look at that in a little bit more detail here. Here we've got regular transcription of one of these microRNA genes. They're relatively small, usually about 100 bases in length. They form a secondary structure. That secondary structure gets recognized by some very specific proteins. Again, it's a secondary structure. It's the structure which is being recognized. It's not the sequence which is being recognized. This is true again. We talked about this for DNA repair, et cetera. So that structure gets recognized. This now gets exported into the cytoplasm. In the cytoplasm, the most critical protein here is the dicer protein. Dicer for chopping things up. What dicer does is it gives you a double-stranded RNA which is between 20 and 25 nucleotides in length with slight overhangs at either end. You see here's a 2-base overhang, here's a 1-base overhang. This then gets separated and put into the so-called RISC complex that we're going to look at in a little bit more detail in just a second. So this is the microRNAs. Small interfering RNAs are exogenous RNAs. So this is double-stranded RNA that's coming from the outside of the cell. And these are usually viral RNAs. And we'll talk much more about double-stranded viral RNAs later on. But this is an RNA which you really don't want. And this process here, which is probably derived from the microRNA regulatory process, basically leads through a very similar process. Dicer chops up this double-stranded RNA into small pieces. One of those pieces gets taken up into the RISC complex, and in the case of small interfering RNAs, this RISC complex chops up the RNA, and chops that RNA very specifically in the middle of that RNA where it's base paired to this small RNA that dicer has actually made. The RISC complex is made up of about 4 different proteins in most cases. The most important of these is this argonaute protein. Argonaute is really the one which is binding to that single-stranded RNA and bringing the two together. Some cases also has a nuclease activity, but argonaute is really the most important one, and I wouldn't bother remembering all of these other ones here. So that argonaute in the RISC complex, the way you get it here again is with drosha for your microRNAs, leading to dicer, again 22 nucleotides. If this has come from an exogenous RNA, so small interfering RNA, you get cleavage of your messenger RNA. If it's a microRNA, then it leads to blocking of translation. Again, either get cleavage of your target or regulation of translation. This all depends on whether you have a perfect match to your RNA sequence or a slightly imperfect match. And so in the case of the microRNAs, you should have a couple of bases that don't match. This will lead to translational regulation, whereas if you have a perfect match, then this leads to degradation. That's the process shown here. Your microRNAs lead to repression. Your siRNAs lead to messenger RNA cleavage. One more aspect of these small RNAs that I wanted to mention, and that is that as well as getting translational regulation or cleavage, you also see chromatin being regulated this way. And that's, again, through the Polycomb proteins that we talked about before. Remember, Polycomb is important for getting DNA methylation to take place. Turns out that there are some other complexes, again, together with argonaut, which will lead to binding of RNA, recruitment of Polycomb complexes, which leads to DNA methylation. And the reasoning for this seems to be that way back when, in the depths of time, when we talked about transposons, remember, most of our genome is transposons, right? We're getting transcription of most of our genome most of the time, both strands. You're going to end up with lots of transcripts from transposons all throughout our genome because they're highly repeated. So you're going to end up with double-stranded RNA, which matches transposon sequences. Do you want transposons to be jumping around in your genome all the time? No. So this is a way to suppress transposons because you're getting the Polycomb proteins and compacting the DNA right around where all your transposons are, so you're not getting transposon expression. So that's another process this is being used for. Finish up, and this is the last slide I'm going to talk about. You can skip the last two, even though they're really important and they might be on MCATs, DATs, etc. later on in your life. But when you have these RNAs, normally when they get exported in the proper process, you've had splicing that takes place. This now is a normal RNA, which will get translated. In many cases, splicing doesn't happen properly, and what you end up with are these exon junction complexes that are downstream of a stop codon. Usually when you translate, the translation machinery, the ribosome, will bump off these exon junction complexes. If these exon junction complexes are not bumped off the DNA, or say the RNA, you arrive at a stop codon, then this is clearly an RNA which is incorrect, and it gets broken down. Now, the way that these things get broken down is, again, through cleavage of the RNA. Why is cleavage of the RNA so important in terms of getting rid of it? If you chop the RNA, that leaves ends. And right at the beginning of this part, I mentioned the exonucleases. If you don't have a cap or a poly-A tail that's bound to the poly-A binding protein, that's all going to get chewed up. So as soon as you have cleavage, then the rest of your RNA is going to get broken down really fast. Again, because of all these exonucleases that are here. So thanks, everybody, for a great term. Don't forget there are evaluations that you can fill out online for this class. And I'll also have some evaluations with the final. So thank you again.